In late 2018, a groundbreaking large-scale comparative study of biological aging across human populations was brought to completion by a multinational consortium of geneticists. This extensive collaboration spanned numerous esteemed institutions, meticulously gathering and analyzing tissue samples from over 48,000 individuals to represent every major regional population on Earth. The overarching goal was to construct a detailed picture of how the human aging process might vary, aiming to pinpoint underlying genetic factors. Initially, researchers anticipated a scenario where the aging process would be largely consistent across all populations, a universal biological clock. They believed any minor variations would be primarily attributed to environmental influences, such as diet, pollution levels, and broader socioeconomic conditions. In essence, their expectation was that any observed regional differences in actual lifespan would primarily stem from life experiences, not inherited genetic variations. However, the data told a profoundly different story. Far from confirming this hypothesis, the findings unequivocally demonstrated the biological clock does not run at a uniform rate. The observed differences were not just minor fluctuations. They were substantial, systematic, and clearly clustered along specific regional and ancestral lines. Even after rigorous correction for environmental influences, the populations exhibiting the slowest cellular aging rates were concentrated in distinct geographic regions of East and Central Asia. This consistent pattern was neither a mere measurement artifact nor an environmental consequence. It strongly indicated something intrinsically embedded within their genetic and regulatory architecture. The consortium meticulously prepared its findings for publication in early 2019, but the subsequent journey to publication proved arduous and politically fraught. By the time the paper finally saw the light of day in late 2020, the most provocative findings had been notably relegated, buried within the supplementary materials. The discussion section had been carefully crafted to deliberately avoid drawing the explicit conclusions that the robust data so clearly supported. Since the late 20th century, human population genetics has been fundamentally structured around a powerful institutional commitment to the idea that genetic differences are inherently minor. Reporting findings of inherited population differences became politically and professionally risky, ensuring the general public consistently received a simplified, managed message. The 2018 aging study stands out as one of the most striking contemporary examples of this pattern. Its findings were simply too clear and robust to be ignored or dismissed within the technical scientific record. Yet, they were simultaneously politically charged enough to necessitate extremely careful management in any public-facing presentation. The data unequivocally revealed substantial, inherited differences in the rate of biological aging between distinct human populations. These differences were systematic, not random, and they distinctly clustered along specific geographic and ancestral lines. Furthermore, the populations that exhibited the slowest aging at the fundamental cellular and regulatory level were predominantly concentrated in particular regions of East and Central Asia. This observation strongly pointed to deep genetic factors carrying substantial functional consequences for human longevity and health. Allow me to describe, in detail, precisely what the data showed, presented in its unvarnished form, before any of this institutional framing was applied. The first major finding underscored that telomere shortening rates differed significantly among populations. Telomeres, for context, are the crucial protective caps found at the ends of our chromosomes. They naturally shorten with each cell division, serving as one of the primary and most reliable cellular markers of biological aging. The 2018 data compellingly revealed that even after meticulous correction for a wide array of environmental and socioeconomic factors, populations of East Asian ancestry consistently exhibited slower telomere shortening rates than the global mean. This was particularly evident in populations hailing from regions such as Honshu, the Korean Peninsula, the Yangtze basins, and the highland territories of Tibet and Bhutan. These differences were far from subtle. Indeed, at their most pronounced, the rates of telomere shortening were observed to be 15 to 20% slower than the global average. The populations displaying the slowest rates of telomere shortening were not randomly distributed, but concentrated in specific geographic clusters. Intriguingly, these clusters showed substantial overlap with regions historically identified in older anthropological literature as the deep heartlands of East and Inner Asian populations. This suggested a long-standing genetic connection. The second major finding delved into methylation age, a more recently developed and highly sophisticated aging marker. This marker quantifies the patterns of chemical modifications on DNA that naturally accumulate as an individual ages, serving as a biological clock. The methylation age data, strikingly, mirrored the exact patterns observed with the telomere data. Across East Asian and Central Asian populations, especially those originating from the ancient heartland regions, methylation ages were consistently found to be biologically younger than their chronological ages, particularly when benchmarked against the global mean. These robust patterns held true across multiple advanced methylation aging algorithms, including the foundational Horvath clock and the newer Grimmage and PhenoAge clocks. This strong consistency across diverse methodologies unequivocally affirmed that the signal was real and not merely an artifact of any single measurement technique. The third significant discovery pertained to mitochondrial function. Mitochondria, often referred to as the powerhouses of our cells, are responsible for producing vital energy. With advancing age, these organelles inevitably accumulate damage, and their efficiency gradually declines over time. The 2018 data strikingly demonstrated that the rates of this mitochondrial decline differed substantially among various human populations. Populations from East Asia and specific regions within Inner Asia displayed significantly slower rates of mitochondrial decline. This finding carries profound implications for cellular energy availability, overall cellular function, and crucially, for resistance to age-related diseases. The groups exhibiting the slowest decline included populations from the highland regions of Tibet, the vast Mongolian steppe, and older heartlands within central China. The fourth major finding centered on regulatory protein expression markers, a cutting-edge set of indicators meticulously developed by the Karolinska team. These markers track the intricate patterns of expression of regulatory proteins, all of which play critical roles in cellular maintenance and repair. The data revealed that East Asian populations managed to maintain more youthful expression patterns of these vital proteins for significantly longer into their chronological age. The Tibetan populations in particular presented remarkable regulatory profiles at age 60 that were strikingly similar to the global mean profiles typically observed in individuals aged 45. These findings suggested something embedded deep within their genetic architecture was actively producing a slower aging phenotype at the molecular level. The fifth and perhaps most crucial major finding served to intricately tie all the preceding discoveries together into a cohesive picture. When the consortium integrated the genetic data, they successfully identified a small, specific set of regulatory genetic variants strongly associated with the slower aging phenotype. Critically, these variants were present at substantially higher frequencies within the slowest aging populations compared to the global mean. These variants were localized within genes known to be involved in fundamental processes like cellular replication, mitochondrial health, and oxidative damage response. These variants themselves were not entirely novel discoveries. Some had been identified in earlier, smaller-scale studies. What was truly groundbreaking, however, was the comprehensive picture it provided of how these variants were geographically distributed and, crucially, how they precisely correlated with the deep ancestral structure of the populations carrying them. The variants displayed a remarkably clear, non-random pattern. In the technical sections, the consortium explicitly identified this source. They called it a Northeast Asian ancient core lineage. This ancient lineage was meticulously traced through analysis of ancient DNA samples extracted from bone fragments, demonstrating its continuous presence in the deep continental interior of Asia for at least the past 40,000 years. This ancient lineage significantly predated the better documented Bronze Age population movements that profoundly reshaped the genetic landscape across much of Eurasia. The genetic variants robustly associated with slower aging were found to be present at their highest frequencies in modern populations that retain the strongest genetic signal of this ancient core ancestral component. The link was unequivocal, but this particular finding, the link between slower aging and this specific, deep ancestral component, was precisely the aspect the consortium chose not to prominently feature in the public-facing version of their paper. Stated plainly, the finding unequivocally shows that this phenotype is inherited, the variants are real, and their geographic distribution is clear. But the implication that there are fundamental differences in biological aging was deemed politically and institutionally too sensitive and charged for the consortium to present in the direct, unvarnished form that the data supported. Consequently, the published paper described these compelling findings in carefully measured and deliberately bounded language. While it did acknowledge population differences and noted geographic clustering, it deliberately framed these discoveries as merely evidence of complex environmental and genetic interactions, whose interpretation was presented as preliminary and whose implications required further research. The underlying technical content of the paper was, in its scientific rigor, honest. However, the framing of these findings was meticulously managed. As a result, the general audience, typically exposed through subsequent press coverage, received a heavily simplified message. They were told the study had identified some interesting variants associated with healthy aging that were worth further investigation. Crucially, the public did not receive the core message. The study definitively documented substantial, inherited differences in the rate of biological aging across human populations. The implications of the actual findings, when stated clearly and without equivocation, are profoundly significant. They suggest that the deep population history of East and Inner Asia, unfolding over tens of thousands of years, fostered and refined a regulatory architecture for cellular aging that is demonstrably more efficient than the global mean. This implies that populations carrying this architecture are, at a fundamental biological level, genuinely longer lived than the standard models of human aging typically acknowledge. Furthermore, these findings strongly suggest that the world's well-documented longevity hotspots, such as the Okinawan populations, the Highland Tibetans, certain rural populations in central China, and inhabitants of the deep interior of Mongolia, are not merely anomalies attributable solely to diet and lifestyle. Rather, their underlying biology has been specifically configured for slower aging through deep ancestral genetic factors, factors that the modern academic establishment has been notably reluctant to discuss in explicit terms. This finding naturally connects to a broader, largely unaddressed picture of buried population history that the consortium implicitly acknowledged. The deep Northeast Asian core lineage is the same component extensively documented in ancient DNA samples recovered from the Altai region, and indeed, it connects directly to the populations that older European cartography labeled as the heartland of territories referred to as Tartaria. The genetic record reveals that the deep continental interior of Asia has been home to long-standing populations whose unique biological inheritance shaped the modern gene pool. The conventional textbook account, portraying these regions as relatively recent migration zones, simply does not account for the profound continuity of this genetic signal. The Tibetan case stands out. They are famous for their high-altitude adaptation, the EPAS-1 variant, allowing them to thrive in low-oxygen environments. That specific variant was inherited directly from the Denisovan population, an archaic human population confirmed from a single, minute fragment of fingerbone discovered in a Siberian cave. The compelling 2018 aging data now suggests that the Denisovan-derived genetic contribution to the Tibetan populations goes much deeper. It may encompass not only the well-known EPAS-1 altitude adaptation variant, but also crucial components of the slow-aging regulatory architecture. The profound implications are that deep, archaic populations imparted a cellular maintenance architecture that actively produces slower biological aging. Mainstream coverage of the Tibetan high-altitude adaptation has been extensive and widely celebrated, fitting comfortably within an acceptable evolutionary narrative. In stark contrast, mainstream coverage of the Tibetan slow-aging architecture has been conspicuously minimal, largely because it defies the sanctioned narrative of human uniformity. Both findings, however, rest upon the exact same rigorous kind of scientific evidence and derive from the same type of comparative genetic analysis. The fundamental difference lies not in the quality of the science itself, but rather in the political and institutional acceptability of the conclusions it unveils. The Okinawan populations of the Ryukyu Islands offer another compelling window into this same overarching picture. Okinawa is globally renowned for its exceptional longevity. Boasting some of the highest concentrations of centenarians in the world, the mainstream explanation has consistently emphasized dietary habits and lifestyle choices. While genetic factors have been occasionally mentioned, they have invariably been de-emphasized in popular and even much academic discourse. The narrative focuses on their unique social structures and beneficial environmental factors, painting it primarily as a triumph of lifestyle. However, when the 2018 consortium data was specifically filtered to include the Okinawan samples within the broader East Asian cohort, it revealed that the slow-aging variants were present at particularly high frequencies within the deep Okinawan founding population. This suggests that Okinawan longevity is not merely a lifestyle phenomenon, it is, significantly, a deep population genetic phenomenon. The well-documented lifestyle factors interact synergistically with an underlying biology that has been profoundly configured by deep ancestral genetics to age more slowly. Similar patterns, compellingly, emerge for other documented longevity populations scattered across East and Central Asia. The Bama County population in southern China, the Hunza populations nestled in the upper Indus Valley, and remote groups in the Mongolian interior all exhibit longevity profiles that the mainstream narrative has predominantly attributed to environmental factors and lifestyle choices. Yet, the 2018 data strongly suggests that the underlying biology in each of these cases carries crucial components of the slow-aging regulatory architecture. Take Bama County. The mainstream explanation for its remarkable longevity has consistently focused on pristine air, local diet, and strong social structures. However, when the data was filtered for rural Guangxi samples, it distinctly showed that the slow-aging regulatory variants were present at substantially elevated frequencies relative to the broader Han Chinese population. This indicates the Bama population is genetically distinctive, carrying a deeper signal of older inner Asian lineages. The Hunza case, located in the majestic but harsh Karakoram Mountains, presents a remarkably similar picture. Famous in Western popular literature for over a century for their purported exceptional health, mainstream academics have often been highly skeptical of their demographic data. But the genetic data stands independent of these demographic claims. Regardless of the precise accuracy of their age statistics, the Hunza population carries a distinctive genetic signal that directly connects to deep inner Asian ancestral components. Significantly, this genetic profile includes elevated frequencies of those exact same slow-aging regulatory variants. Whether the population truly lives to extreme ages at the rates claimed is one important question. However, whether the population possesses a biological inheritance inherently expected to produce slower cellular aging is a completely distinct question. The genetic data from the 2018 study definitively answers this second question in the affirmative. The inheritance is real. Regardless of how the demographic question regarding Hunza's actual extreme longevity is ultimately resolved, the populations of the Mongolian interior offer a third compelling case study. The deep rural populations inhabiting central and northern Mongolia, particularly those maintaining strong connections to ancient nomadic herding traditions, exhibit genetic profiles that robustly retain the strongest signals of the deep Northeast Asian core lineage. The 2018 data revealed these populations carrying the slow-aging variants at frequencies among the highest documented in any modern human population. Intriguingly, their lifestyle factors are not conventionally considered particularly favorable for longevity. Their diet is typically high in animal fats and proteins, the climate they endure is exceptionally harsh, and their medical infrastructure is often limited. By virtually every conventional measure of environmental influence on aging, the Mongolian interior populations should theoretically age rapidly. Yet, when their actual biological aging profiles are measured directly, they reveal precisely the opposite trend. These populations age more slowly. This striking divergence underscores how genetic inheritance, in this context, effectively overcomes significant environmental headwinds. The slow-aging architecture, intrinsically embedded within the regulatory machinery of their cells by deep ancestral genetics, continues to function effectively, seemingly regardless of the surface-level lifestyle factors that mainstream literature has historically emphasized. For anyone endeavoring to understand their own biological inheritance, what this ultimately means is that the profound ancestral history of the populations inhabiting East and Central Asia includes a unique configuration of regulatory genetic variants that collectively produce measurably slower biological aging. These variants are undeniably real. The resulting slow-aging phenotype is also real and demonstrable. Their geographic distribution is clear and non-random. And, fundamentally, populations that carry the strongest signal of this deep ancestry possess, on average, a biology that ages more slowly than the global mean. In the years since the completion of the 2018 study, the consortium has remained conspicuously quiet regarding these specific, far-reaching implications. The lead researchers have simply continued their academic careers, smoothly moving on to other safely funded projects. And the raw data? While technically available for any specialist willing to download and analyze it, it has not been widely publicized. It sits quietly on servers, mostly ignored by the broader scientific community. The published paper has indeed been cited within the technical literature, but always in carefully bounded and often understated ways. Crucially, the most profound implications have not been pursued in any major public-facing forum. Instead, the textbook version of human aging continues to stubbornly emphasize uniformity across populations and the overwhelming, almost exclusive influence of environmental factors like diet and exercise. The deeper, more complex picture, one in which inherited differences in the rate of biological aging cluster along specific deep ancestral lines and persists, confined almost entirely to the supplementary materials. It is accessible in principle, but practically buried. It is never fully assembled or clearly presented for the general public. The very public whose own biological inheritance is, ironically, being described in those hidden pages. This is, regrettably, the standard pattern for how such potentially controversial findings are managed. Within the institutional scientific framework, stability and the maintenance of political consensus often outweigh disruptive truths. The data is diligently collected, the analysis is meticulously performed, but the framing is aggressively managed for public consumption. When examined honestly and thoroughly across populations, the aging of the human body is simply not uniform. The deep populations of East and Inner Asia demonstrably carry a regulatory architecture that facilitates slower aging. This architecture is a direct inheritance from ancient ancestral components that have been continuously present in the heartland of the continental interior for tens of thousands of years. Furthermore, this unique architecture connects directly to the broader, largely buried population history of these regions, including those territories that older historical sources collectively termed Tartaria. The mainstream academy has, by choice, opted not to discuss these profound findings in the explicit terms that the compelling data so clearly supports. Yet, the data is readily available for anyone willing to genuinely look. The conclusions are there for anyone willing to draw them, and the populations whose biology has been profoundly configured for slower aging by these deep ancestral genetic factors continue, on average, to live longer and healthier lives than the global mean. This holds true regardless of whether the official narrative chooses to acknowledge these underlying biological reasons. If this is the rigorous investigation you came here for, you know exactly what to do. Subscribe to the channel, share this video, and keep watching because there is much more to come. The aging genome is just one chapter. The deep population history of Inner Asia is another. And the buried biological inheritance of the East Asian populations is yet another. We are working through them one at a time. To truly grasp the magnitude of this biological inheritance, we must look back to the crucible that originally forged it. We must return to the last glacial maximum, roughly 25,000 years ago, when massive ice sheets covered much of the northern hemisphere. While most other human populations naturally migrated south toward warmer, more forgiving climates, the ancestors of the Northeast Asian core lineage remained trapped in the deep continental interior, the mammoth steppe of ancient Siberia. In this environment of unimaginable harshness, absolute caloric scarcity and extreme cold acted as a brutal evolutionary filter. A standard human biology characterized by rapid cellular turnover and high energy waste simply could not survive here. Survival required a fundamental biological restructuring at the very molecular level. The mitochondria, the cell's powerhouses, had to become hyper-efficient, generating maximum heat and energy with absolutely minimal resources. Simultaneously, the body needed to drastically slow down its overall cellular degradation to preserve vital functions over longer freezing periods. This is the true, hidden origin of the slow-aging regulatory architecture identified in that 2018 study. It was not developed as a mythical fountain of youth or a magical secret to living past 100. It was an absolute, unforgiving necessity for surviving the most extreme and deadly epoch of human prehistory. The genetic variants for slow telomere shortening and youthful protein expression were literally forged in the ice. When the ice finally retreated thousands of years later, these populations carried this extraordinary biological engine with them. They expanded outward, taking this optimized, hyper-efficient cellular architecture into the broader Asian continent, shaping history forever. As the glaciers retreated, this hardened population dispersed, carrying their hard-won biological inheritance into the fertile valleys of East Asia. This ancient core lineage became the foundational genetic bedrock for modern East Asian and Inner Asian populations. However, as populations expanded and intermingled over millennia, the concentration of these specific survival variants didn't remain perfectly uniform. In certain isolated pockets, highland plateaus, remote valleys, and specific island chains, the genetic signal remained incredibly concentrated and pure. But it is fundamentally incomplete. You cannot simply eat your way into an Ice Age biological architecture. To admit that the foundation of their longevity is inherited would shatter the egalitarian consensus of modern biology. It would require acknowledging that human populations are simply not biologically identical blank slates. So the 2018 study remains buried, and the lucrative lifestyle industry continues to thrive, selling a half-truth to a desperate public. The answer reveals exactly why the mainstream blue zone diet advice is fundamentally flawed and dangerously misleading. Evolutionary biologists refer to this phenomenon as an evolutionary mismatch. We are conditioned to seek endless abundance. Yet for the descendants of the Ice Age crucible, unchecked abundance is literally toxic. The 2018 study didn't just identify a few interesting variants. It inadvertently exposed a suppressed chapter of human history, a history where surviving the most brutal epoch on Earth meant developing a hyper-efficient, slow-aging cellular architecture. The academic establishment, however, continues to ignore this complex reality, and they let a multi-billion-dollar lifestyle industry sell you the blue zone lie, pretending a diet can rewrite thousands of years of evolution. This channel exists to unearth precisely this kind of science, the rigorous, controversial research they won't teach you. If you are ready to question the comfortable consensus and dig into the raw, uncomfortable truths, subscribe to the channel, share this video, because the next chapter is already being written.